It's 2 o'clock on the dot. We only have a few attendees online at this point, so let's just give it another minute or two. Thank you all for being on time, and hopefully in just a moment we'll have some more folks joining us. All right, everyone, let's go ahead and get started. Thank you so much for being here. If your calendar is anything like mine and anything like Missy's, I know that you all are just as busy as we are. It is that time of the year, that January through March time period is so busy for everyone locally. So thanks for taking the time out of your day to join us for the Indian River Lagoon um, National Estuary Program's What's Up in the Watershed. Each month, we do a webinar about projects going on in the Indian River Lagoon. As we always like to say, every project equals progress. And so we just try to highlight the different projects that are happening with our partners and interesting things that we know about that we'd like to share with you. And if you know about any interesting projects that you'd like to share with us, please feel free to give us suggestions for future webinars. Today's webinar is being presented by Missy Weiss, a native New Yorker whose love for nature and the sea began at an early age. She has her MS in biological oceanography from Stony Brook University's School of Marine and Atmospheric Sciences, as well as a BS in marine science from Long Island University, Southampton College. Or Missy, am I supposed to say Long Island?
Yes, you
Okay.
are.
Additionally, she received graduate level certification specializing in secondary education. She currently has marine experience in science research, environmental education, and of course, citizen science program development. Her role as ORCA's Director of Citizen Science and Education allows her to combine all of those amazing areas of interest and to provide meaningful opportunities for citizen science and to participate in ORCA's applied science research. And this project is particularly exciting. This project is ORCA's One Health Fish Monitoring Project that collects data on toxins and toxicants in fish from not just the Indian River Lagoon, but also surrounding waterways. And by involving citizen scientists, ORCA continually expands this data set. And I cannot say enough about how open they are with all of the data on their website is absolutely amazing. And I'll put in a little plug, and maybe you can talk about this at the end, Missy. Also, the data jam that ORCA hosts, all of this data that Missy's going to be showing us or sharing with us today is available on their websites and might culminate in other interesting projects. Missy, thank you so much for being here today. Um, I got to help dissect a mullet the other day so I could see what the experience was like, and I'm excited to share this with everyone else. So when you're ready, you could go ahead and share your screen, and I'll turn my video and audio off.
Great. Thank you so much for having me. I'm excited to be here to talk a little bit more about ORCA's One Health Fish Monitoring Citizen Science Project. It really is a privilege and an honor to be able to present to all of you about this really impressive and exciting project that I hope you all get involved with in some way or another. So for those of you who are unfamiliar with ORCA as an organization, first and foremost, it stands for the Ocean Research and Conservation Association. We're a local nonprofit, and while we work up and down the entire Indian River Lagoon, our headquarters is in Indian River County. And our mission since 2005 has always been to the dedication to the protection and restoration of aquatic ecosystems and the species those waterways sustain. And we do this in several ways. We do this through our innovative technology, our science-based conservation action, and community education and outreach. And specifically, we try to uphold this mission in four major ways. So as I mentioned, innovative technology is really what ORCA was founded off of or based off of. With regards to technology, we have our real-time water monitoring devices, also known as Kilroys. We currently have the largest real-time water monitoring network in the Indian River Lagoon, if not in the state of Florida. And so all of that data is accessible on our website, teamorca.org, at any point. We also uphold the mission through our applied science research. And under that, we really focus in on ecotoxicity as well as One Health issues. We do this through our citizen science, so the engagement and education of community members, people of all ages, backgrounds, ethnicities, religions, in participating in real world science. And lastly, we do it through our education. So we make it a point to not only collect valuable, unbiased, reliable information, but also be able to teach others about the importance of that collection and what it can do to drive change. So we work with students of all age, from preschool all the way up to our adult learners, because we really are all lifetime learners, or at least should be. Most of our efforts at ORCA are focused on preserving the Indian River Lagoon. Obviously, this waterway is the lifeline of our region. Economically, it brings in billions of dollars annually to our area, and ecologically, we know its importance there as well, being deemed at one point the most biologically diverse estuary in the entire United States. Now, as I mentioned, we're an applied science research organization. And so what that means is that we really try to make it a point to look at the problems impacting the lagoon, and there are many, in a holistic fashion. So we try to collect as much information as we possibly can to look at those problems and how they are interacting or how they may be impacting each other. And there's no better way to leverage that than incorporating people in the community in the research through citizen science. So currently at ORCA, we have 4 citizen science projects that people can participate in. One is our land to sea monitoring. That's really our habitat restoration work. We also have our pollution mapping citizen science project, One Health Fish Monitoring, which is the topic of today's conversation, and then our student-centered project, which is really a stepping stone for students to enter into the world of citizen science, which is a day in the life of the Indian River Lagoon and Lake Worth Lagoon.
Missy, could I pause you for just one moment? Aaron, some folks are still seeing your screen, so I want to make sure that everyone can see Missy's screen. Aaron, did you change? Okay, I think we're good to go now, maybe.
I hit the
Okay,
stop share
perfect.
button.
Thanks, Aaron. Okay, sorry about that, Missy. Go ahead.
No problem. Yes, of course. So before we jump into the project itself and the details of the One Health Fish Monitoring Project, it's really important to know what the term One Health really means. And so One Health is an idea, it's an approach, and a recognition that the health of humans is connected to the health of animals and our shared environment. So in other words, when the environment's health is impacted through contamination or pollution, it can have impacts on human and animal health and can lead to disease and health concerns. And when we protect one of these sectors, we ultimately protect all of them. What this approach also recognizes and what it calls for are collaborative efforts across multiple disciplines. So those people working locally, nationally, and globally in order to attain optimal health for people, animals, and our environment. And again, so that's going to require communication, coordination across public health, veterinary, and science sectors. And so where we really see ourselves fitting into this is collecting the needed information and data to inform the other sectors and to drive change. So specifically under this umbrella is ORCA's One Health Fish Monitoring Project. And what this citizen science project looks at is it investigates the transfer of pollutants. It investigates the transfer of toxins and toxicants into the Indian River Lagoon and those freshwater canals entering into the Indian River Lagoon, and determining, well, what are the pollutants that are entering into the Indian River Lagoon, and are those pollutants accumulating into fish that have been identified through angler surveys as being fish that are commonly consumed by people. And then once we have that data, we're able to estimate the potential human health risk, if any, through the consumption of those fish. Never with the guise of telling people not to consume fish, but rather providing information and data to make decisions based off of that data that is collected, which ultimately can be shared with stakeholders and decision makers to help with water quality, as well as advise human health consumption guidelines if required. So we've had a lot of success with the One Health Fish Monitoring Citizen Science Project so far, which is really lovely, in that we've had over 300 active citizen scientists engaged in this project to date. And I think one of the reasons why it's been successful is there's lots of different ways that people can get involved in this project, however they see fit. So one way that you can get involved in One Health Fish Monitoring is that you can survey anglers. So if you recall, when I talked about kind of the topic of this presentation or the objective of the project, I mentioned that we obtain the list of fish that are recognized as being highly consumed by humans through angler surveys. So we're always looking for people to go out into the community, popular boat ramps or fishing locations, and survey those individuals who might be catching those fish for consumption. Another way that people can get involved is that they can catch and donate their legal and in-season fish to the project. And yet another is that you can come into our Center for Citizen Science, or we often offer pop-up processing locations where you can dissect those fish. So as Heather mentioned, about once a week or so, we welcome volunteers into our lab and we will process, so remove those tissues and organs from those fish that are later analyzed. And then yet another way you can get involved is that we can teach you how to run some extractions. So you can actually be the scientist who is involved in following a series of standard operating procedures or a recipe to extract those toxins from those fish tissue. So again, there's lots of different ways that people can get involved in this particular project. Now, currently, and this list is always growing or ebbing and flowing, if you will, in that this is a list of current analytes that we test for on all of the fish tissue that are donated to the project at this point in time. And when I mention, say, tissue, I'm really specifically talking about the fillet. So again, if we're trying to estimate potential human health risks, then we want to be able to analyze those parts and pieces of the fish that people are consuming. So right now we are looking at biotoxins, so specifically microcystin. This is the toxin that is being produced by microcystis or a cyanobacteria. We also have looked for heavy metals, so mercury, lead, and cadmium. Every fish tissue at this point in time is analyzed for total mercury. We look for glyphosate. This is an herbicide found in products at one point like Roundup or other herbicides. microplastics, per and polyfluoral substances, also known as PFAS. So we work with the University of Florida as a collaborator to help us analyze tissue samples for those compounds. We're currently trying to develop the methods of extraction and detection in UV filters. These are the compounds that are found in sunscreens. And so we're really focusing at this point in time on looking at oxybenzone. That is one of those compounds that have actually been banned in some areas of the country and world. And so we're focusing there. So is that UV filter entering into our waterway and into our marine food web? And then finally, we were fortunate enough to receive an Impact 100 Martin grant last April, and that will now allow us to analyze at least 150 fish from Martin County for a whole panel of pharmaceuticals and controlled substances. So as I mentioned, this is kind of the current list. Different emergent contaminants of concern can be added, or as funding allows, may be subtracted. So for any of you on this webinar right now who are saying to yourself, man, I would really love to donate my catch to that project. That sounds really interesting. This is the current list of species that have been identified as being those that are consumed by people. And again, when we're thinking about consumption, we're not just thinking about an individual eating the fish, but we're also talking about, well, who does that particular angler share that fish with? Are they sharing it with their pregnant wife, their young children, their elderly parents? How often are they eating those fish? And so again, these are the target species up at this point in time. And luckily enough, we have been able to partner with a lot of local bait and tackle shops and nonprofits who have agreed to act as drop-off locations. So if you live in Brevard County and you don't want to necessarily drive your fish to our Center for Citizen Science, which is in Vero Beach, you could drop off your fish at one of the Brevard County bait and tackle shops. And at this point in time, we have one in at least every county that we do this project in. So Volusia all the way down to Marin County. And again, if you don't want to catch and donate your fish, but you want to get your hands dirty and participate in the processing, then again, we offer that training workshop at least once a month, and then opportunities for you to practice and hone in your skills at least once a week. So this is a great way to get involved in the project. Or again, if you want to learn how to do microplastic analysis or other extractions, we welcome you to join in. So at this point in time, I want to share with you some preliminary results for the project. As I mentioned in that list of analytes that we currently study, there's a lot that we do, and all of that data is accessible, but I'm just going to kind of highlight a few of these pollutants at this time. So let's kind of start with even just donations. So at this point in time, you can see that most of our fish are coming from Indian River County at about 470 and Brevard County, likely due to the proximity to our Center for Citizen Science. But we have been really focusing in on obtaining fish specimens from the southern portion of the lagoon, primarily Martin County. And we've done a really good job over the last couple months of increasing that number, but we're always welcoming new fish from these regions. So this is just kind of to set the stage. While this is very important and we analyze a lot of our data by county, we also like to hone in and really focus on specific waterways. So for example, this is a picture of St. Lucie County. And within this St. Lucie County map, we have color-coded or divided this county into different sectors. So you can see I'm kind of outlining SLC3, that's one sector. This is another sector. And so within those sectors are either popular fishing locations or specific areas. And so once we get enough fish from these different sectors, we're able to not only analyze that data at a county level, but also at a sector level. So if you're an individual who says, well, I often consume fish from SLC 13, what does that data show? We can hone in on that area specifically, which is helpful. So let's start with microplastics. So most of you are familiar with microplastics. I'm sure it's in the news all the time because truly microplastics are ubiquitous in our environment, in animals, and in our own bodies as humans. And microplastics are just small plastic pieces categorized as being less than 5 millimeters, which have been identified as being harmful pollutants to our ocean and aquatic life. And studies are indicating that these microplastics not only bind to these pollutants, so therefore they're a vector to the pollutants into our environment and into different biota, but unfortunately they also cause known health damage. So that could be oxidative damage, DNA damage, changes to our gene activity, and even cancer development. So microplastics are certainly something that people are studying, as well as are being incorporated in this study as well. So when we talk about microplastics at ORCA as part of our One Health Fish monitoring project, the way that we ultimately get that data is that once a fish is donated to the project, and again, every fish that we have ever analyzed has come from a donation, they're then processed by a volunteer. Those fillets are then homogenized or ground up by volunteers. That sample is aliquoted, and then it is digested and filtered, again, all by citizen scientists. And we're even at the point now where we have volunteers who are trained well enough to count those microplastics on the fibers. We currently look for microplastics in both fish stomachs and tissues. And we're able to look at this data by location in the lagoon, microplastics type, microplastic color, and size categories. We're not refined enough. We don't have the equipment that allows us to tell which specific plastic it is. But for us, at an applied science research approach to it, we're comfortable with being able to give this version of the data. So we've been able to analyze 428 independent or individual fish stomachs. Of all 428 of those fish stomachs, 71% of those stomachs have at least one piece of microplastic present in that stomach. For fish fillet, so again, the part that people are consuming, we've been able to analyze 540 specific species or samples of fish, and 60% of those tissues have had at least one microplastic present in that analysis. When we look at microplastics by type, we're really looking at 4 categories. We're looking at whether that microplastic is a fiber. So fibers are going to come from places like ropes, lines, sails, film, which are really kind of thought of as plastic bags or food wrappers, fragment, which is just little pieces of plastic. Think of a water bottle or a pail that has degraded and broken down over time. And now here you have this little fragment of plastic. And then granules, which are also kind of referenced as spheres. And those are the plastics that are kind of melted down to create other plastic products. So from our pie chart here, you can see that nearly 3/4 of all the plastics that we've been able to identify fall under that fiber category. And when we break it even down further to see if there's any differences in the fish by different counties, so Brevard, Indian River, St. Lucie, and Martin, you can kind of see quickly that overall the trend is all the same, and that fiber really is the predominant microplastic type in fish, no matter what county that you are in. We can also look at microplastic by color. That often helps kind of narrow down maybe the sources of those microplastics. And again, this is just kind of the groupings that we've created based off of what our data is showing, but we have categories in blue, black, red, green, and other captures all of the, obviously the other colors of microplastic as well. And you can see that about 40% of all the microplastics are blue and about 1/4 are black. So there's some speculation as to why blue might be the dominant color. Some people have suspected that it might be attributed to all of those tarps that we wrap our boats or trailers or usually in close proximity to the lagoon. And as they weather and break down, they could be attributing to some of what we're seeing. And then once again, looking at the color by location, again, you can see across the board, blue really is that dominant fiber, followed by black. So that's kind of the microplastic data that we have at this time. We're continuously adding new data to that site. So even what I just showed you is a little outdated, but know that every 6 months we offer a findings to date to our active volunteers, but also new volunteers or people who are interested, kind of like what we're doing today, where we show the most up-to-date and current information about what we're doing in this project. And so that next one is at the end of the month, and you can tune into teamorca.org again to kind of sign up to get on the mailing list to receive that. Let me move on to glyphosate next. So again, glyphosate is an herbicide. It's actually the most commonly used herbicide worldwide. And glyphosate has some contradictory information surrounding it. So in other words, the EPA summary review of glyphosate indicates that it's unlikely to be a human carcinogen, whereas the World Health Organization says that it's probably carcinogenic to humans. And so because of this contradictory information, that's one of the reasons why we're really keen on monitoring it, not only in our fish, but also in our water and our sediment samples across all of our projects at ORCA. And what I can clearly say about this particular compound is that it certainly is persistent in water and sediments, and spoiler alert, our fish. So it is, or at least I should say, it was an active ingredient in products like Roundup. I was actually just sent the last safety data sheet of the newest version of Roundup 4, and glyphosate is not the active ingredient at this time. And so I haven't personally done enough digging, but I just read that safety data sheet yesterday. So if that is the case, that's a good thing. But what we do know about glyphosate as a chemical is that there are biological impacts, not only to animal health, fish health, but human health. And so we have been able to test for glyphosate. The way that we do that, again, is with the help of all volunteers. Once we get that homogenized tissue, that fillet that's been ground up, we're able to run an extraction on that and then run an enzyme-linked immunoabsorbent assay, or an ELISA. That generates a concentration. And so we've been able to run 570 fish and test them for glyphosate. And what we've found is that 99% of all the fish tested have a presence of glyphosate in their fish tissue. Now, this figure is representing average glyphosate per county in parts per billion in all fish from that respective county. So for example, if we look at Martin County, we have analyzed 88 individual fish from Martin County for glyphosate and have created an average. And that's what you're seeing here. So we've got county on the Y and concentration on the X. So at this time, we see that Volusia fish have higher average concentrations of glyphosate, but please note that that sample size, it's just 8 fish. So when I ran statistics on this, I'm not going to be including Volusia at this point in time. I did run an ANOVA though on the other counties, and at this point in time, Brevard County fish have higher average glyphosate concentrations compared to Indian River, St. Lucie, and Martin County. A lot of people are then curious about, well, how does that look in specific species? And so what you're looking at here is now a breakdown of average glyphosate per species, where we had at least 10 individual species to test. So for example, we have species on the Y with our 2 freshwater species on top. And then we have other brackish water species further down, with concentration on the X, again, in parts per billion. So you can see when we look at the Indian River Lagoon fish, black drum, spotted sea trout, bluefish, snook kind of are at the top of those concentrations, where Atlantic croaker and catfish are a little bit lower. Again, we're talking about parts per billion. So when we're talking about high, that's relative, right? So black drum has about an average of 0.3 parts per billion in when we calculated an average for those 15 fish. I do want you, though, to keep in mind this black drum number here and the concentration of 0.3 parts per billion, because when we compare that glyphosate concentration to established human advisory levels that are already in place, I want to kind of walk you through what we're seeing. So again, in order to make that comparison, the first thing we need to do is we took that fish species that had the highest average glyphosate, which is black drum, at 0.3 parts per billion. In order to convert it to that advisory level, the first thing we need to do is some math. So we need to convert parts per billion to milligrams per kilogram. And when we do that, we get 0.0003 milligrams per kilogram of glyphosate in black drum on average. Now what we also need to do is we need to know, well, how much does Joe here typically consume when he eats fish or black drum? And so based off of our angler surveys, the typical serving size is 8 ounces. And we also need to know Joe's weight. And so we're going to take the average weight for males, which is 180 pounds. And we can now calculate how much Joe is consuming if he were to have a serving of 8 ounces of black drum with regards to glyphosate, which is 0.00007 milligrams. So is that a lot? Is that a little? What does that even mean? So that's where we kind of compare that data to what's been already established as far as acceptable daily intake values. So this ADI is different depending on where in the world you live. So in the United States, that ADI for glyphosate is 1 milligram per kilogram of body weight per day. Whereas in Australia and New Zealand, it's a bit more conservative. They only have an acceptable daily intake of 0.3 milligrams per kilogram of body weight per day. And so if we were to compare our black drum to this acceptable daily intake, we would allow ourselves, if we lived in at least Australia, which is more conservative, we'd be allowed to have 24.5 milligrams of glyphosate. In order to reach that with regards to black drum, we would need to consume over 350,000 servings of black drum per day, which is not happening and would frankly be quite disturbing. But what we have to recognize here is that while our glyphosate concentration in our fish are low, that is just one route of exposure to glyphosate at this point in time. It could be through other things that we consume in our diet. It could be through our interaction with the environment. It could be in our drinking water. So again, this is one route of exposure of us acquiring glyphosate in our own bodies. Is it acceptable that there's glyphosate at all in fish? Not in my opinion, but at this point in time, our data is indicating that it is not at a concentration that is meeting or exceeding our acceptable daily intake. Heavy metals is the last analyte that I wanted to just share with you a little bit about. Not everything that we do is doom and gloom. We learn a lot from what we do. And so we have tested cadmium as a heavy metal at one point on a subset of our fish species. And we were able to do that with the help of a small grant from the Indian River Lagoon National Estuary Program. Because what that grant allowed us to do is just to determine whether or not cadmium at all is entering into the marine food web from the Indian River Lagoon. So we tested 100 fish, and with that information, we were able to determine that just 2 of those 100 fish came back with any detectable concentrations of cadmium, which is a good thing. And so we no longer invest our time or our money into looking at cadmium because we have a good subset of data here. That's not the same story of what we see with total mercury. For every fish at this point in time that has come into the project, we've tested for total mercury. And again, 99% of all those fish detected have some level of mercury in their fish tissue. If we look at this the same way that we did for our glyphosate, we'll break it down by county. But before I do that, I think it's important. I always get the question, well, where is this mercury coming from? Most of the mercury that's entering into the Indian River Lagoon is coming from atmospheric deposition. So it's getting caught up in those winds, and then when it precipitates, it's allowing that mercury to fall into the environment and then accumulate over time. And so, unfortunately, a lot of the mercury that's getting incorporated into our marine food web locally is coming from overseas. So it's not even a local source necessarily. But when we look at total mercury by county, the same way I did that with glyphosate, where we take every fish that's been analyzed and we create an average and standard error. This is kind of the information that we're seeing at this point in time. So we're seeing again now counties on the X, concentration of total mercury is on the Y. This time it's in parts per million or milligrams per kilogram. And we can see that pattern is slightly different than what we saw for glyphosate. Now what we're finding is that Martin County fish have significantly higher concentrations of average mercury compared to St. Lucie, Indian River, Brevard County. I did not include Volusia in that assessment, again, because of the small sample size. So that is one of the reasons why we were so keen on trying to better understand the pollutants entering Martin County because of this level. Now, mercury has current human health advisory levels in place. That's not the case for some of the other analytes that we look at, such as PFAS. But for mercury, we can compare it to established guidelines. So this orange line that you're seeing running across all of the bars here is the Florida Department of Health's advisory level for women of childbearing age and children. And the green line is indicative of a joint advisory from the EPA, FDOH, and FDA for the general population for total mercury in fish tissue. And so again, at this time, we're seeing that on average, the fish across the board are exceeding that advisory level of women of childbearing age and children. And Martin County fish are meeting, and in some cases exceeding, that joint advisory level for the general population. So this is, again, why we're doing what we're doing currently with the help of our citizen scientists, is to kind of track and monitor this over time. When we look at it with respect to species, same idea in that we're seeing that different species accumulate different concentrations of mercury. And so what we're finding at this point in time is that there are really 4 species that are exceeding that current advisory level for the general population. And those include bluefish, crevalle jack, ladyfish, and spotted sea trout. But there are also species of fish that don't meet or exceed any of those or either of those advisory levels. So species like striped mullet, Atlantic croaker, black drum, which we just talked about, cichlids and crappies, they're at a level that are falling below that first advisory level that we discussed. And so again, if you are a subsistence fisher who relies on the fish from our waterways to not only feed yourselves, but your family members, and you rely on that fish for a solid protein source, then potentially this data can help you make decisions as to what species or waterways you may be wanting to consume those fish from. And then I just wanted to highlight in this graph, it's the same exact one, the one that we looked at, but now we're starting to also have enough freshwater species. And so I know that there's a lot of freshwater anglers out there. And so I just wanted to kind of highlight where we're finding at least 3 of those species in our data. And probably one of the most exciting things is I really appreciated that Heather said that a lot of the data with regards to this project is online. We really make it a point to share and disseminate all of our data, but specifically with this project, you can find it on our website. You can find recordings from our previous findings to date. And then very, very soon, we're going to be debuting a brand new citizen science data site, and we're going to start with the fish project. And what this data site will do is it will allow our data to be interactive. So I wanted to just show that to you quickly, because hopefully in April, which is Citizen Science Month, we will open this up to the public, and you too can kind of play around with the data specific to this project. So when you first enter the page, you'll be greeted by this site right here, where it'll kind of give you an overview of fish counts, but then you can play around with other data sources. So if I'm interested in PFAS and I want to look at that with regards to our fish, I can do that. I can see average PFAS totals by county, or I can even look at it by species. And so you'll have a chance to kind of toggle through the different analytes that we're able to share with you, so that you can know more about what are the pollutants accumulating into our waterways and being incorporated into our marine food web. So
Wow, that is amazing. When you kind of showed us that just a moment ago, I didn't know what you were getting at. That is so cool. Thank you for sharing that.
well, you're welcome. We're excited to be able to do that with everybody. And I should state that it's live. So every time I add new data to that, the back end of our data set, this information will change. So even from what I just showed you today, we've now processed this morning fish. And so that fish count number will be different from what I shared with you even in this presentation today. So it's constantly live and being updated as new information is being brought to it. And then, so I just want to end with conclusions and kind of summarize some of the data that I shared with you today. First is with regards to microplastics, we're seeing that blue is the most common color, while fiber is the most common plastic type. This also jives with some UCF data in the water samples and oyster samples. So we're seeing it also in our fish. We know that glyphosate is ubiquitously found in fish, but at very low concentrations at this time. And we know that mercury concentrations vary geographically and certainly by species. So as we look ahead in this project, we are going to compile our samples and send those samples off to Clemson University so that they can be analyzed for pharmaceuticals and controlled substances, specifically for Martin County fish. We're always looking for funding to be able to incorporate other fish from other counties. So that's on the horizon. We're going to be evaluating our data to really start honing in on specific species. So narrowing down that list even further in areas. And then always open to the idea of exploring new emergent contaminants of concern. Unfortunately, that is the nature of our world right now, in that there are endless pollutants. And so we want to be able to be on the cutting edge of looking in that for our local waterway. And then obviously we look forward to allowing others to play with data and utilizing our citizen science data site, because science is fun and we can learn a lot from it, obviously. And so last but not least, if you do want to donate to this project, I would just say that there are protocols that you must follow. This is all on our website. Moral of the story is the fish has to be kept whole, on ice or frozen. It must be legal and in season. We need the name of the angler, your information, the date you caught that fish, and the location. So if you can provide latitude and longitude or some kind of marker to denote where you were, that's all we need. And last but not least, when you do drop off those fish at a local bait and tackle shop, give us a heads up and let us know that you did so that we can pick them up right away and get them processed and ready to go. Finally, I just want to thank all of those volunteers, those community citizen scientists who have donated their time and their effort, got their hands dirty, whether it's donating those fish, processing those fish, or helping us extract. Couldn't do any of it without my colleagues, as well as our interns, as well as our academic collaborators at Embry-Riddle, UF, and Clemson University, as well as all of our funders, because science is expensive and we need people who trust in it to donate to it, and we appreciate you. So with that, I would be happy to take questions or let you all digest what I just said.
There's a lot to digest there. One thing I'm curious about is, will the new website be ready for folks who want to participate in the data jam? And can you tell us a little bit about the data jam?
Yes, thank you for reminding me about that. The new website likely won't be ready yet for Data Jam, but luckily on our website already, there is a project page that provides the data sets that people can use. And so what Data Jam is it's a competition that merges science and art. And so typically when we think about displaying or communicating science, a lot of the times we think of that being done in the written form through a research paper or journal article. But there's lots of ways to communicate the story of data. And so Data Jam is a competition for students and adults. We provide our ORCA citizen science data sets to the public, and we task them with analyzing that data and then interpreting it in an artistic way. So for example, one of the data sets that we offer people to look at is our microplastics data. And so one of the students that won 2 years ago, actually it was a group of students, created an interactive video game where they were the diver, and every time they touched a fish, that fish would show, which was an actual fish that we processed, would show the number of microplastics, the type of microplastics, and that at the end of the game, it would always come out the same way, in that the dominant type was fiber and the dominant color was blue. And so that was a really creative and unique way. We've seen sculptures, we've had crochet items, which I can't do. We've had songs and drawings and murals and so much more. So it just kind of puts a creative spin onto data interpretation.
I love it. And tell us what the date is of when that competition will be
Yeah,
announced.
perfect.
Sorry, I'm
No,
putting
that's
you on the
okay.
spot.
I have it. Um,
Okay.
so the final showcase is on May 8th. It's going to be held at the Heritage Center this year. It is free and open to anybody, and we will precede that showcase with a talk about the merge of art and science by our CEO and a local artist. So people are welcome to join there. And you would have to have your project submitted by the end of April. And so the best way to learn more about that and kind of register and sign up is to check out our website and that specific project page to learn more about the Data Jam.
And I did put that specific link in the chat if folks are interested. One of our friends from Space Coast Surfrider Space Coast chapter, Chris Baker, her project was just so amazing. And so it's a really cool thing, and I'd love to see even more adults getting involved with it.
Yeah, thank you for
It-
that.
It's
That's love.
very cool. A few questions coming in. How does ORCA's data influence public awareness and community action regarding conservation?
Yeah, that's a really great question. So we've kind of been in the position where we've been trying to generate a large enough sample size to be confident in our data so that then we could be able to go and have those conversations with Florida Department of Health or our local level, to be able to have those conversations with the EPA and share the information that we have. And so we've been very diligent to make sure that we have a large enough sample size across all counties, as well as good species representation before we have that conversation. We're very close to that point now, which I'm looking forward to. And at that point, that's where that data will be have discussed and handed over. We've also shared our data with obviously UF because they helped generate that PFAS data, but currently Florida does not have a PFAS advisory level in fish. So we are optimistic that that data will also be provided to those agencies to help generate that advisory level for our state. So that's kind of the way that we foresee this data set driving change.
I think you kind of answered two questions in one. Another question that came in, how can findings from the project influence environmental policy and regulations?
Yeah, and I kind of alluded to that in
Yeah.
the first response is, and again, it's about being not pointing fingers. It's not about being alarmists. It's about making sure that the data is in a place that is reliable. We do appropriate QA, QC, making sure everyone's following the right standard operating procedures and protocols so that when we do have those conversations, there's a trust. And so that is a really important component to all of our citizen science work, but work as a whole. So I'm optimistic that that will lead to policy change.
Here's another one. What measures can be taken to reduce pollutant levels in the Indian River Lagoon? And I don't know if this person means, at the homeowner level or perhaps even at the municipal level, if you maybe could answer both.
I mean, there's so many things that we can do, but I think the first thing is kind of doing what this person just said is, well, what can I do? What can I do to help? What can I minimize? And so I've been really having this internal conversation with myself lately about and recognizing that compromise is important, right? So in my mind, I want everyone to be able to do everything that you could to minimize your impact, less plastic, making sure we're always bringing our water bottles, keeping our septic, if we have that, up to date and at the highest technology, and so on and so forth. making sure that our grass clippings aren't being blown into the canals or drainage, being mindful that if we are using some kind of application on our lawn, what that is. But compromise is important, and I think that people need to do something small if they're not sure of themselves, so that they can realize that, hey, this is easy, this is achievable, I can make an impact. And then you'll really challenge yourself to do more. So I think there's lots of things that people can do to minimize their impact, but plastic is a huge source of pollution, and it's not just the plastic itself, but because it binds to other pollutants, it really becomes a vector to other ways to drive that pollution. And so that would be a good place to start, as well as what are we doing on our own lawns that could minimize the amount of nutrients or pesticides or herbicides that are entering into the waterways.
I'm surprised that it doesn't surprise me that fiber is the dominant plastic type found, but I am surprised that other sources of plastic aren't in similar numbers, considering how plastic breaks apart so easy, plastic litter breaks apart so easily. Were you surprised by that?
I didn't really know what to expect, honestly. I didn't. And I wonder whether some of that is just because of the ability to discriminate by the fish. Have the ability, those gill rakers are there for a reason. So if it's too big, they can kind of spit it out kind of situation. But when I think about fiber, again, that's coming from sails, ropes, but our clothing. And so think about how much lint is in your dryer every single time you change that. And that is not what's being caught up in your washing water and then sent down the drain. So I am, but I'm not. And our wastewater
Right.
treatment plants aren't at the capabilities at this point in time to remove some of that. And so that is another big avenue of advocacy, is to make sure that we are supporting improvements to our wastewater treatment plants.
I read recently that in Europe, all new washing machines actually have to be fitted with, and not aftermarket, but at the time of sale, a microplastics filter on washing machines. Do you know much about that yet?
I don't. I know you had mentioned that when I saw you a couple of weeks ago, and I was curious about that, but I haven't read anything about that personally yet. So again, any step that people are willing to take to make a difference, it will help.
And I know I can't, I've never used one myself, but I know that in the States, we can access aftermarket filters that we could put on our washing machines. And I've been trying to read up on those to see, if they're effective, if it's worth it, what are the challenges. And it seems that space is a real challenge. Having enough space between the wall and your machine itself to actually install these aftermarket ones, as opposed to when a machine is constructed with them in the first place, it's flush with the back of the machine.
Yeah, it's a good idea.
Very interesting. I have a couple more questions here. Maybe we have time for one more. Um, mm, how does the project address potential biases or even gaps in the data collected?
Hmm. I'm not 100% sure I understand what this person is talking about, but I guess what I would say is, um, one of the ways that we try to minimize those gaps, again, is trying to be as comprehensive as we can. And
Mhm.
that takes time. So those surveys are really, really helpful at generating, again, not only what species we should be targeting, but also providing that human health dimension. Now, a way that that could be biased is, are we uh, gathering those surveys from a good representation across the lagoon? So like, are we getting into all the different communities to be able to get good representation? That is something that we're chipping away at, but I'm sure there are areas that we haven't been able to pinpoint. So that's where partners like Bethune-Cookman have been very helpful, because they're filling in the gap of the northern portion of the lagoon in Volusia County, which we don't really have, that's 2 hours away from here. but they're able to start filling in some of the gaps to better understand what is the population of individuals in that area of the lagoon. Is it different than what we're seeing in others? So we can kind of help overcome those biases in that way. We do have very strict QA/QC. So after every 10 samples, we run duplicates or replicates on every single thing that we
Mm,
do.
mhm.
To make sure, again, that because sometimes when you engage citizen scientists, there is question, but the amount of rigorous training that we provide, again, is there and in place to make that the data is trustworthy and is not biased in any way.
So that'll be perfect for our last question. When is the next training date for fish analysis, specifically interested in microplastics?
Okay, cool. Um, Great question. So we have a fish training tomorrow.
Oh boy.
Yep. Tomorrow afternoon at 1 p.m. And then we have more opportunities on March 24th, which is a Monday. And probably the best thing to do to know other dates would either be to just email me personally and I can provide you with those dates and times, or you can check out our website because we have a citizen science calendar that lists the upcoming training opportunities. And
Great.
you'd be expected to register. And then the microplastics is a little bit different. We usually start you with the processing, and then we get to know each other, and we date a little bit, and then I will train you on how to do the microplastic analysis.
I am going to put the link. Ah, why is the chat has not. Oh, there we go. Okay. I'm going to put the link to the calendar. I've put several links. They're all basically at teamorca.org, but you can get signed up for their e-news. You can learn more about their data jam, and you can look more in depth at that citizen science calendar. I've put 3 links in the chat, but they all start at the same place. So if you can't remember all of these by the time we finish up today, and you'd like to just go to teamorca.org. It's a great website, very user friendly. They can find all that. Oh, and there's Missy's email as well.
Yeah.
Missy, thank you so much. Let me just click on the questions to make sure no more open questions at this time. You did great, and we kept it right at an hour, so that's perfect. Thank you for joining us. Thank you everybody for participating in the middle of what is a very busy season. I sure appreciate everyone's attendance, and I always love learning more. Yeah.
Thank you.
So thank you, Missy. Thank you, attendees. And then don't forget, if you have some area of interest that you might like to see discussed in a webinar, let me know, and I'll see if we can make that happen in the future. And otherwise, just follow us on our own social media. That's where we try to post, everything that we've got going on in the moment. We also have our own newsletter if you'd like to get signed up for that. And I always have to put a plug in for our awesome Snook license plate. All of the extra funds collected from that stay in the counties where those license plates were sold. And by law, all of that money has to go to restoration projects. It can't go to anything else. So multiple ways folks can get involved beyond today. And we are recording the webinar, and we will get that posted as well. Thanks, everybody.
Thank you, Heather.
Awesome job. Thanks,
Thank you so
Missy.
much. Appreciate the opportunity. Bye,
I'm
guys. Have a
sure.
great day.
Thank you. You good, Aaron?